angiosperm life cycle, as well as sexual reproduction in angiosperms. And as we'll see, the reproductive and menstrual cycle when we get to female humans here towards the end of the lecture. And you'll really want to pay attention to that in particular, potentially for, you might see a prep quiz that has a lot of questions on that cycle in particular. So first one I want to, first we'll talk about plant reproduction. A lot of this may be familiar to you because, we did see a lot of this in earlier chapters, but it's going to cover this in a lot greater detail. So try to keep track of, certain vocab terms and whether they, are in a haploid or diploid phase, as well as whether or not they undergo meiosis or mitosis. And then lastly, you want to see if it kind of corresponds to male, organs or female organs, the analogs in plants. So let's take a listen to this one first.
Angiosperms are plants that reproduce by means of flowers. Flowers are showy, colorful structures made of modified leaves. A flower usually contains both male and female reproductive structures. A flower can have up to 4 types of modified leaves: petals, sepals, carpels, which consist of a stigma, style, and ovary, and stamens, which consist of an anther and filament. Ovules and megasporocytes are present inside the ovary. Let's review the sequential stages of the angiosperm life cycle. The process that occurs in the ovary, which initiates the development of female gametophytes, is the division of a megasporocyte by meiosis. Inside an ovule, the megasporocyte, 2N, undergoes meiosis to form 4 cells, but only one of these cells survives as the megaspore, N. The nucleus of the megaspore divides by mitosis 3 times, resulting in 1 large cell with 8 haploid nuclei.
You might want to remember that.
Membranes then grow between these nuclei to form a multicellular structure called the embryo sac, which is the female gametophyte. The large egg cell is located at one end of the embryo sac. A diploid cell within the anther divides by meiosis to form 4 microspores, N. Each microspore, N, divides once by mitosis to form 2 cells. A tough wall is formed around the outside, creating a pollen grain, which contains a male gametophyte. Pollen grains are transferred by wind, water, or an animal to a sticky stigma in a flower of the same plant or another individual of the same species. After a pollen grain lands on the stigma, it forms a pollen tube that elongates downward through the style into the ovary. 2 sperm nuclei, N, form inside the pollen tube. The pollen tube enters the ovary and releases the 2 sperm nuclei into the embryo sac. 1 sperm nucleus fertilizes the egg, forming a diploid zygote. The other sperm nucleus fuses with 2 nuclei in the center of the ovule to form a triploid 3N cell that will develop into a nutritive tissue called endosperm. This process is called double fertilization. The triploid cell divides rapidly by mitosis to form endosperm, a tissue rich in starch, lipids, and proteins. The zygote divides by mitosis to form a sporophyte embryo with a rudimentary root and 1 or 2 cotyledons, seed leaves. The integuments of the ovule become a tough seed coat.
This term cotyledons is where we get the term monocot versus eudicot. They say, the video just mentioned that it'll form 1 or 2 cotyledons. They're just the first leaves that appear on the plant. And if only 1 appears, then it's a monocot, and if 2 appear, it's a eudicot.
With the embryo and endosperm inside. As the ovules develop into seeds, the ovary surrounding the ovules develops into the fruit, which will protect and help disperse the seeds. The fruit falls to the ground and seeds germinate. A seedling uses the food stored in the endosperm and cotyledons to grow until it can begin photosynthesis. The young plant grows into a mature sporophyte that produces flowers, and the cycle continues.
All right, so yeah, a couple things to make note of there. You know, the flower has both male and female components, and then when fertilization occurs, either with itself, depending on the flower, or with individuals from another species. What's important to note is that this term double fertilization refers to those sperm cells, each two sperm cells penetrating something. One of them penetrates to form the embryo, which is a diploid structure, as well as the endosperm, which is triploid, which is going to serve as the early nutrients for that developing embryo until it gets large enough to kind of fend for itself and become its own plant. So some vocab terms you want to be familiar with. And so this has done a good job of like breaking down the general life cycle, but you'll also want to learn more specifics about reproduction, specifically in these flowering plants, the angiosperms.
The dominant plants on Earth are the angiosperms, the flowering plants. Flowers, the reproductive structures of angiosperms, produce fruits, which enclose the seeds. A flower is generally composed of 4 parts: sepals, petals, stamens, and 1 or more carpels. Although the sepals and petals can play an indirect role in reproduction, for example, attracting pollinators, here we'll concentrate on the stamens and carpel, the parts directly involved in reproduction. The stamen, the pollen-containing structure, is composed of 2 parts, a stalk-like filament and an enlarged anther. Inside the anther are chambers containing diploid cells called microsporocytes. Each microsporocyte undergoes meiosis to produce 4 haploid microspores. These microspores divide by mitosis to form male gametophytes, or pollen grains. The immature pollen grain consists of a small generative cell enclosed within a large vegetative cell called the tube cell. The generative cell will later divide to form sperm. When conditions are right, the anther opens to release the pollen grains. Pollen is produced in large quantities because most of it will not reach a receptive flower. Some falls to the ground, some is eaten by insects, and so on. The carpel is made up of 3 parts: the stigma, the style, and the ovary. A carpel may contain more than 1 ovary. The stigma at the top of the carpel is specialized for receiving pollen. The stalk-like style supports the stigma. At the base of the carpel is the enlarged ovary. The ovary contains developing ovules. After fertilization, the ovary becomes the fruit. Each ovule contains a single diploid megasporocyte and is surrounded by a continuous covering, except for a narrow canal at one end called the micropyle. The megasporocyte undergoes meiosis to produce 4 haploid megaspores. 3 of the megaspores degenerate, leaving a single functional megaspore in each ovule. The megaspore divides by mitosis 3 times, producing a total of 8 nuclei, which segregate to form 7 cells. The most important elements of the female gametophyte are the egg, located at the micropyle, and the 2 polar nuclei. The polar nuclei are found in the largest cell of this structure, at the center of the female gametophyte.
So notice that there are multiple cells within this megasporocyte structure, but these are going to be the 2 of the 8 total that are going to be really important. And these are the 2 cells that will essentially get penetrated by sperm cells in that double fertilization
Largest
step.
Cell of this structure, at the center of the female gametophyte. Sexual reproduction in angiosperms involves the transfer of pollen from an anther to a stigma, a process called pollination. In self-compatible flowers, the stigma is receptive to pollen from the same flower. Self-incompatible flowers require pollen from a different plant. In both cases, pollen can be dispersed by living vectors, such as insects, or non-living vectors, such as the wind. In this example, pollen from the anther of one flower is transferred to the stigma of another flower. After the pollen grain lands on a receptive stigma, it germinates, producing a pollen tube. The pollen tube grows down through the tissue of the style. At some point during its journey, the generative cell of the pollen grain divides by mitosis to form two sperm nuclei or male gametes. The pollen tube continues to grow until it reaches the ovary. It then enters an ovule through the micropyle. It ruptures one of the cells next to the egg and discharges the two sperm nuclei. In most plant groups, fertilization is straightforward. One sperm nucleus fuses with the egg to form a diploid zygote. In angiosperms, however, an unusual event called double fertilization takes place. The other sperm nucleus fuses with the two polar nuclei to form a large triploid, 3N cell. The triploid cell resulting from this second fertilization begins a series of mitotic divisions that forms a tissue called the endosperm. The function of the endosperm is to store nutrients.
All right, so I think this provides some greater detail into how reproduction in flowering plants occurs. So I think it's worth watching and maybe even going back on this video and re-listening to this a couple of times because, you know, I understand a lot of vocab terms are thrown your way here, but, you know, try to apply one of those learning techniques to grasp each of those concepts. Maybe use a concept map, for example. This is just reiterating some of those points, how you'll also see the term embryo sac, which I don't think actually appeared in the video, but it's the same thing as the female gametophyte in this scenario, and it forms all inside of a tissue called the megasporangium. This is reiterating these same points, but again, note that eventually through mitosis, 8 nuclei are going to form, you know, and essentially 6 of them are kind of, you know, play supplementary roles, but there are 2 kind of main cells in this, those polar nuclei in the cytoplasm, which get fertilized by one of the sperm cells to form the endosperm, and then another cell that kind of gets designated as the egg. And then in terms of males, it's, you know, a little bit simpler, a lot less to learn about because essentially all they're doing is transferring themselves to the female structure where, you know, the next generation is essentially going to be made. The male gametophytes, similar to in animals, are essentially providing genetic material. But some terms that you want to kind of be aware of are, you know, when we're dealing with males, you'll see, you know, these microsporangia and microspore terms come about. And the important thing to note about this is that within the microsporangia, you start off with diploid cells, and they're undergoing meiosis to produce microspores. So remember, spores are haploid, just like gametes. Gametes will fuse to form a diploid zygote, but both spores and gametes are haploid. So whenever you see the term spore, think haploid. If you see sporophytho, it's a diploid thing that makes a haploid spore. And then just some further terminology. We talked about those, or we saw the video about the 2 sperm cells that end up fertilizing the female, but they actually have specific terms, the generative cell and the tube cell. And so be familiar with those terms as well. And then pollination is essentially what the last portion of the first video talked about, how pollination is essentially that transfer of pollen to some receptive stigma. So remember, it's all going to start on the stigma, where the pollen then forms a pollen tube down the style, think of it like a stalk, and then ultimately makes it to the ovary. And then, you know, just reiterating this point about double fertilization. It's called double fertilization because of these 2 sperm cells penetrating 2 different cells within the female to form the endosperm and then the actual egg fertilization.
The function of flowering is to produce seeds and disperse them to locations where the young plants are likely to survive. At each step, there is an emphasis on protecting the offspring. This activity reviews some of the most important steps. Events in seed formation begin with pollination, in which wind or animals bring pollen to the flower. The growth of pollen tubes cause the flower to discard its colorful parts, petals and stamen. After pollen arrives, the bright petals are a hazard. They attract animals that might eat the delicate young fruit. We've enlarged the ovary and sliced it open. An ovary contains one or more ovules that will become seeds if they are fertilized. One ovule is shown here, with the front removed to reveal its parts. The ovule is attached to the ovary wall by a stalk that provides nutrients. Starting from a pollen grain at the stigma, which is not shown, a pollen tube grows down through the carpel to the ovule. It carries two sperm that enter the ovule. One sperm fuses with the egg cell to make a zygote, which is the first cell of the embryo. The other sperm fuses with the two nuclei in the central cell, making a triploid nucleus. The triploid cell will grow and divide to make a food-storing tissue, the endosperm.
All right, so that's essentially the gist of plant fertilization and the plant life cycle, and we'll go into a lot more detail of this tomorrow in class. But for the last half of this supplemental lecture, I want to focus on animals. So specifically chapter 30 focused on plant reproduction, and 36 will focus on the animal side of reproduction. And for animals, it's important to note that animals can undergo both asexual as well as sexual reproduction. So we're probably associating animals with sexual reproduction because that's the only way we can reproduce. But animals like the sponge and many invertebrates can also reproduce asexually. And so the main differentiation between them from a scientific point of view is essentially where sexual reproduction involves, requires this fusion of a male gamete, the sperm, and the female gamete, the egg. So keep in mind, these gametes, the sperm and the egg, are both haploid, and they fuse together to ultimately form a diploid zygote. So the zygote is going to be that first stage that becomes diploid. Once you have, when you're just dealing with the sperm and egg separately, those are haploid. And then asexual reproduction kind of foregoes all of this. And it's pretty much the simplest form of reproduction where it just doesn't require the fusion of an egg and a sperm. But it can happen multiple different ways. And we'll see an example of one animal, the hydra, which is in the phylum Cnidaria, one phylum we learned about a couple modules ago now. And you'll see that the hydra can basically do it all. It can reproduce both asexually and sexually.
The Cnidarian hydra can reproduce both asexually in a process called budding and sexually by the production of egg and sperm. The hydra in the opening scene of the video is growing a young bud close to its base. Buds form as an outgrowth of the two-layered body wall. At first, the tentacles are relatively short, and the gastrovascular cavity of the parent and the young bud are continuous. As the bud reaches maturity, the tentacles elongate, and the gastrovascular cavity becomes separate. Eventually, the bud pinches off and drops to the substrate to begin independent life. Credit courtesy of Graham R. Kent and.
So, this is just one way of reproducing asexually, this budding off process, where a small part of you just kind of pinches off. Another common way is you'll see the term fission up here. So fusion is the fusing of two similar size, two cells. And fission is specifically describing this kind of like budding, but essentially you're getting two similarly sized individuals result from it. So the main difference really between budding and fission is fission produces two similarly sized individuals, and budding is where just a small piece of you and pinches off to become a separate organism. So with, animals in general, and especially humans, we have a vested interest in learning about our own reproductive system. And this is going to reflect on the prep quiz as well. You'll see a lot of questions about understanding the human reproductive system in particular, because by knowing our reproductive system, we can kind of use that to apply it to other animals as well. You gain a benefit by knowing about your own reproductive system, but know that we are all kind of evolved from a common ancestor, and so we see everything we see in humans, we see kind of synonymous versions of this in other animals as well.
The reproductive system of the human male consists of testes, various ducts and glands, and the penis. The brain, acting through hormones, controls the process of reproduction. Sperm develop in the testes in coiled tubes called seminiferous tubules. Spermatogenesis produces several hundred million sperm each day. The sperm from each testis mature and become motile in a coiled tube called the epididymis. When a man is sexually aroused, muscular contractions propel the sperm out of the epididymis and into the vas deferens, which loops over the bladder. The vas deferens joins a duct from the seminal vesicle to form the short ejaculatory duct, which with the ejaculatory duct from the other side joins the urethra. The urethra conveys the sperm out through the penis. In addition to the seminal vesicles, the prostate gland and the bulbourethral glands contribute to the seminal fluid that nourishes and protects the sperm and lubricates their path.
And then let's see the female side of this.
We will combine our discussion of the ovarian and menstrual cycles to show how hormones coordinate the activities of the ovaries and the uterus. The brain controls reproduction, acting via hormones produced by the pituitary gland. While a woman is menstruating, her hypothalamus begins secreting a releasing hormone, which stimulates the anterior pituitary to secrete FSH and LH into the blood. FSH, follicle stimulating hormone, stimulates the growth of an ovarian follicle in the ovary. The follicle consists of a developing egg cell surrounded by cells that nourish and protect it. The developing follicle begins secreting estrogen, a female sex hormone. Estrogen shapes development of the female reproductive system and female secondary sex characteristics such as broad hips and development of the breasts. Its immediate role is to stimulate regrowth of the lining of the uterus in preparation for possible pregnancy. Low levels of estrogen also exert negative feedback on the hypothalamus, keeping blood levels of FSH and LH relatively low. Basically, what is going on here is that the brain triggers development of an egg, and the follicle containing the egg signals the uterus to prepare to support the development of an embryo. Negative feedback assures that only one follicle develops at a time.
If you're not familiar with this term of negative feedback, all it's really essentially meaning is that something kind of inhibits something else instead of promoting, greater production of it, which would be a positive feedback. A negative feedback is where a compound like estrogen, binds to a certain site in the brain and then inhibits the production of LH, luteinizing hormone, and FSH, follicle stimulating hormone. So the production of one compound results in the reduction of the production of these two other.
As the follicle grows, it secretes more and more estrogen. This stimulates further development and thickening of the uterine lining. Estrogen level reaches a peak just before the midpoint of the cycle. Now response of the hypothalamus and pituitary to estrogen reverses. The higher level of estrogen actually stimulates the hypothalamus to signal the pituitary to secrete a burst of FSH and LH.
So you can see how complicated it becomes. So depending on the phase, this, what I referred to as a negative feedback, turns into a positive feedback, where now the more estrogen there is, the more FSH and LH there's going to be. So it goes from a negative feedback to a positive feedback.
It is the burst of LH that triggers ovulation on about day 14 of the cycle. The ovum develops, the follicle ruptures, and the nearly mature ovum is released from the follicle and swept into the oviduct. LH, or luteinizing hormone, also causes the ruptured follicle to develop into a glandular structure called the corpus luteum. After ovulation, the corpus luteum continues to secrete estrogen and increasing amounts of a second female hormone called progesterone. Both hormones contribute to further thickening of the uterine lining in preparation for possible pregnancy. Estrogen and progesterone also exert negative feedback on the hypothalamus and pituitary, suppressing FSH and LH secretion.
So now we're back to that negative feedback. So it goes negative feedback to a period of positive feedback, which triggers ovulation. And then once ovulation occurs, progesterone gets made, and these two make a negative feedback. So negative, meaning more of it, results in less production of these other two, FSH and LH.
This keeps additional follicles from developing after ovulation. We can summarize ovulation and the following events like this. When the follicle is nearly mature, it signals the hypothalamus to trigger ovulation. The ruptured follicle, now called the corpus luteum, prevents additional follicles from developing and signals the uterus to prepare for implantation and development of an embryo. What happens next depends on whether the ovum is fertilized or not. If the egg is not fertilized, the corpus luteum has a short lifespan. Suppression of LH after ovulation causes the corpus luteum to degenerate. As the corpus luteum's output of estrogen and progesterone drops, two changes occur. The lining of the uterus begins to slough off, the beginning of the menstrual period and start of the next cycle. The drop in estrogen and progesterone also reduces feedback inhibition of the hypothalamus and pituitary. This triggers secretion of FSH and LH, which stimulates the development of a new follicle.
So, up on this, until this point, we've seen that more estrogen results in less production of these two hormones. But the flip is also true. If there's less estrogen, then that means that it's not inhibiting the production of these hormones as much. So a negative feedback loop can mean that more production of one hormone results in less production of the other, but also the flip, less production of hormone A, in this case estrogen, results in more production of FSH and LH.
So if fertilization doesn't occur, the corpus luteum degenerates. Without its hormones, the lining of the uterus breaks down and a new follicle starts to grow. If the egg is fertilized, it starts to develop and implants in the wall of the uterus. The embryo secretes a hormone called human chorionic gonadotropin, HCG for short. HCG functions like LH to signal the corpus luteum to continue its secretion of estrogen and progesterone. Later, the placenta takes over this function. Progesterone and estrogen maintain the lining of the uterus and suppress the development of any more follicles. In effect, the embryo signals the corpus luteum to stick around, maintaining the uterus for embryonic development and stopping the monthly cycle. Human reproduction is complicated. It depends on precise coordination of the ovaries and uterus in response to hormonal signals orchestrated by the brain.
And so one last thing that I'll mention here before I end this, since we're at about half an hour into this supplemental lecture and I don't want it to be too long, is really familiarize yourself with the hormones involved as well as the steps, both in terms of what the steps actually are, as well as when they occur within the menstrual cycle. So in your textbook, you'll see figure 36.12. Be kind of aware of what's going on in each phase of the reproductive cycles, particularly of females. So breaking it up into those follicular, luteal phases, and in the middle, ovulation, and know when they occur, because that, these are questions that might pop up. And so, yeah, I will leave this at that for now, so it's not too long. And I will